This lecture is on nucleic acid metabolism. We already covered carbohydrate metabolism in very much in extent. Then we looked at the lipid metabolism, also amino acid metabolism. Now also going to consider nucleic acid metabolism. Basically, nucleic acid is not about the whole nucleotide, that is the phosphate, the sugar, ribose or deoxyribose, and also about the base. So it's not going to be looking at the making of the nucleotide as the three components, but rather only focusing on the making of the base itself. Here, given the understanding between the salvage pathway at the top and then the de novo pathway at the bottom there. In terms of salvage pathway, it is basically recycling the old base, and then with the help of pentose phosphate pathway, which generates the ribose 5-phosphate, and then by from ribose 5-phosphate converting to become phosphoribosyl pyrophosphate, referred as abbreviated as PRPP. It can be then possible to used in the making of new nucleotide by using recycle base. So that's salvage pathway. Salvage pathway is good because it doesn't need so much of energy, unlike that of de novo pathway, where you can see it still requires the activated ribose of PRPP, which comes from pentose phosphate pathway, but also need other donors such as amino acid, such as ATP needs a lot of activation because this is a biosynthesis pathway, and also in terms of carbon dioxide, which is already been incorporated as bicarbonate ion, and also other members such as the tetrahydrofolate. So for the biosynthesis of the base, we look at the pyrimidine biosynthesis first. That is the one ring structure, and then we will look at purine biosynthesis, the two-ring structure. In terms of de novo synthesis of pyrimidine, it all starts by having the ribose sugar attached at the end rather than from the beginning. For the purine, it will come from the beginning itself. And then in order to build the members of the one ring structure, it requires the contribution of bicarbonate. Bicarbonate ion will provide carbon, and aspartic acid will provide a number of members, either nitrogen or carbon. And then you have the ammonia that provides the nitrogen member, but usually it is not by free ammonia, but rather by the donor of glutamine, the nitrogen member itself. So this slide summarizes the making of pyrimidine, that is UTP as well as cytosine triphosphate. So I just want to clarify, it starts off with the 3 steps, that is the utilization of bicarbonate, ammonia, and then with 2 ATP, all 3 steps happening in one single enzyme to form carbonyl phosphate. That means these 2 members of the pyrimidine structure is contributed by carbonyl phosphate. Then comes the aspartate, the amino acid, to provide the remainder of the members, carbon as well as nitrogen. And then only comes PRPP to form the full nucleotide structure. It will first form UTP, and then from UTP it's possible to form CTP. And then not been covered yet, but later in this lecture you will see from UTP, uracil, by putting a methyl group, it will then form TMP. Okay? And then also from CTP, it's possible to convert it to deoxy CTP with the use of NADPH as the reductant. Now we look at the purine biosynthesis, which is making of the two-ring structure, that is ATP as well as GTP. Okay? But unlike pyrimidine, here purine biosynthesis, from the beginning itself, the sugar and the phosphate comes in and it stays on until some 11 steps before it can form the two-ring structures. So just to highlight again the fact that it's possible to make purine either by de novo pathway as well as that of the salvage pathway. So this slide summarizes the whole thing in terms of making the purine ring structure, where you can see up here the contribution done by many donors in order to make the two-ring structure. So in terms of amino acid, you have aspartate, you have glutamine, you have again another glutamine here, and glycine coming together to provide certain members of the purine ring structure. Then you also have the tetrahydrofolate here as well as here, contributing to the carbon in two locations, and also bicarbonate to form the carbon here. And not to leave out, you also have the PRPP from the beginning to be linked up to the building of the purine structure at the nitrogen. So what you have after many steps, you will finally get inosine monophosphate. And then you already see how from inosine monophosphate can form GMP and then eventually GTP. And then from, we didn't see, but from IMP also can make ATP. And then to ensure that from RNA you can also form the DNA, these ATP as well as GTP will be the ribose sugars will be converted to deoxyribose in order to obtain deoxy ATP as well as deoxy GTP.